Yes, so about talking about consensus, your lab has, you and your team have been focusing a lot on two areas, development of vaccine and trying to be able to come up with sensitive and specific diagnostic tests. I would like us to look at the first, the work on the vaccine development against onchocerciasis virus. I think this was part of now Dr. Robert Adamu's
Okay.
PhD. So in this project, you collaborated with Professor Subgi at ULB in University Libre de Brussels, and also Professor Remini Rose, who is also at the
Yeah,
Vrije University Brussels. Can you
right.
tell us a bit about this multi-epitope vaccine that you've been working on?
Yes, so it's a vaccine. Multi-epitope means there are many epitopes in the protein. Most vaccines have been developed just from a single protein
Yeah.
and with some few epitopes. So what we have decided to do is now to get epitopes from many proteins and then fuse them together because
Yeah,
these epitopes.
with yeah, with epitopes, you mean where these where things that are, or maybe some proteins that are available on the microfilaria that
Yes,
immune
so
system can
definitely.
recognize. Yeah.
Right. So definitely a vaccine is a protein from the parasite itself presented to the immune system of the patient, and the patient is going to produce antibodies against it. And what is there is that if a parasite presents one protein today to the host will generate antibodies to it.
Yeah.
The next day, the parasite is not presenting the same protein. It is going to present another protein to the host, and the host will now need time to develop another antibody to this to this uh, to this protein. So what is there is that we have now taken many parts of the parasite and put together so that no matter which part the parasite presents to the host, we already have antibodies which
Against it.
will be able to bind and kill the parasite. That's what we call multi-epitope. So we have taken so many parts of the proteins, which we call epitopes. We have put it in a single protein, now multi-epitope antigen, and that is what we are going to use at vaccine. So it will present so many epitopes to the host, and the host will be able to produce many types of antibodies at the same time, so that even if the parasite comes with any of those, presents any of those proteins, the antibodies are already there, present. That's what multi-epitope antigen stands for.
That is quite an innovative approach to develop to producing a vaccine against oncocerciasis virus.
Yeah.
Are there some vaccine candidates that have been tested in the clinic or that are also preclinically tested? And when you compare them to the OVDRK DKR2 that you've tested or that you've generated, what are the advantages of what you have in terms of efficacy over what is already available in literature?
Okay. For now, there are just two uh, uh, vaccine candidates which are currently being tested, but at the preclinical level, meaning that they are tested in animal models, and this
Yeah.
is OV RA2 as well as OV103. These are the two antigens which are currently being tested. And these antigens are not multi-epitope antigens. They are single antigens, meaning that the number of epitopes they carry will be fewer than the number of epitopes our own antigen carries.
Yeah.
So we definitely have an edge over them that our own will have a wider spectrum as compared to these overall 2 and OV103. So our own has a wider spectrum than
Yeah.
theirs. Although the OV103 and RA2 have gone a further step ahead and is at the level of the preclinical studies.
Yeah, great. Can you give us a brief of the outcome of the results you've obtained so far with the vaccine in terms of experimental work done so far?
Yes, we have had some indications already that our antigen can elicit some protection, because when you have an antigen, you have to test some parameters to see uh, if it can really protect. And we find that when the antigen is injected, they elicit some responses which are protective to
Yeah.
the host, and the parasites may not get established, because we have molecules which are produced like interferon gamma, like interleukin 2, like cytotoxic T cells and
Cells.
cytotoxic helper cells. So when we have these responses, then we have indication that our molecule is will be protective against the parasite. So there are strong indications that the antigen, the multi-antigen, the multi-epitope antigen will be protective.
Yeah, that is quite interesting. I will of course be looking forward to the outcome of the vaccine. Also, you've been investigating the diagnostics of Onchocerciasis volvulus in another project. I think that it was the PhD of Dr. Kabiru Shinto.
Yes.
Um, you design a chimeric diagnostic marker for diagnosing Onchocerciasis volvulus.
Yeah.
What is a chimeric marker? I would like to understand first what a chimeric marker is, and can you give us a bit of enlighten us about the project?
Yes. So chimeric means you have two proteins or three put together. It's a chimera.
Mhm.
So it is in the same way as having many epitopes that are put together. So chimera means proteins which are have been brought together, linked together to serve as a marker. So it's a chimera, meaning that if that protein, if you detect it in an individual, it means the individual is infected. That's a marker. So if we see OVMAIN1 somewhere in the human, then we are sure that that individual is infected with onchocerciasis.
Yeah, so we have some tools that are already being applied in the clinic for diagnosis. I remember when I did my when I was practicing back home from during the clinic, we used to do skin snip to diagnose microfilaria. Apart from skin snip, are there other serological tests available that are now being used, or because why do we need a new diagnostic marker for
Okay.
onchocerciasis volvulus?
Okay, we need a diagnostic marker first, because when you it could the onchocerciasis can be detected just by palpation to see if the nodules are present or not.
Yeah,
But what is happens is that some of the nodules are deeply rooted, such that even you palpate, you will not be able to palpate those nodules.
yeah.
So you need a diagnostic tool to indicate that there is a nodule sitting far beneath the skin, and it should that's an indication that the nodule is present. The second is by microscopy, which you have said, but microscopy is not sensitive enough, because the skin snip which you cut might not contain the microfilaria,
And it's
which
quite
you want.
and it's quite invasive.
It's quite invasive. People don't like that their skin be cut. And if the microfilaria load is not very high in the body, that skin snip may not even have it.
Mm.
Another method is by PCR, because you can detect the parasites by polymerase chain reaction. But it is found that this test, although sensitive, is it needs a trained individual who can handle it, and it is not cheap. So if you want to test by PCR, many people will not be ready to do it because of the cost, and you also need specialized scientists who can do that. Another test, which is now serological, is by testing for OV16 antigen.
Mhm.
So when you test for this OV16 antigen and you find it, then you are very sure that the person is sick. But this has only been possible in the children, and not in adults. So you can OV16 works for children, but and not in adults. And secondly, not even all the children respond to it. So the test is not 100% sensitive. That's why
Uh,
you need some more, you need more diagnostic tools in order to test for this infection.
okay. Yeah, just for clarification, why is OV16 used only in children, rather not also in adults? Are there some?
Yes, OV16 is used not used in adults because it is an antibody test. It has been found that the antibodies to OV16 stay for too long. So you can
Mm,
even have eliminated onco, but antibodies for OV16 are still present, and they will be telling you are sick.
yeah,
Whereas,
positive.
but
Yeah.
for children, it lasts for a very short time. So it is
Okay.
very easy for you to know that okay, there is infection or there is no infection. That's for OV16.
How will you compare the so far? How will you compare the specificity and sensitivity of this OV Mani one diagnostic marker, the chimeric diagnostic marker that you've developed with the OV16 test in terms of sensitivity and specificity?
When we tested, we found that the two are having the same sensitivity. Also, the sensitivity of OV16 as well as our own protein were almost the same.
Yeah.
But we did further was that we we could uh, we could test for OV16 and also test our own antigen main one, and we noticed that if we combine these two tests, that's screen for OV16, screen for main one,
Mhm.
when we find that we will have a 100% sensitivity and 100% specificity. So we propose that these two tests should be done in order to so first with OV16, second with our own test in order to confirm. And we found that when we do those two tests, we have the sensitivity and specificity increases abundantly.
And indeed, this is very innovative and promising because if you don't have a good diagnostic, it becomes very difficult to treat, and also it
Yes,
becomes very complicated to eliminate.
right.
What sometimes I always feel bad, or it saddens me because sometimes we have promising candidates or drugs or some development, but sometimes they or usually they fail to be transferred or valorized into things that can be commercially available. With this promising technology that you've developed, are there some what are the plans forward? Are there plans forward for valorization of the OV money one?
Yes, our plan is to because the problems people are facing very much in our own medium is the problem of cost.
Yeah.
So what we intend to do is to move ahead and have what we call a rapid diagnostic test, so that if you have a drop of blood, a drop of urine, a drop of sweat, you just put on it, and then you have the results on the spot. And this will be very cheap compared to the PCR test, the ELISA test, which is and so on. So we feel that we are going to move to a rapid development of a rapid diagnostic test and it will solve the problem of invasiveness. It will solve the problem of cost, because it should also be field applicable, that if
Yeah.
we take the field, we can get our results in time. It's also field friendly, because it's user friendly, sorry. It's
Yeah.
user friendly. People find it easy to use than going to be carrying PCR machines, getting yourself involved in complicated experiments. So we intend to move forward with this to the development of a rapid diagnostic test, which people will find it appropriate to use. It will be cheap for them, it's affordable. We think that that is our next step.
Yeah, great. I'm happy to hear that. So 2020, so 2025 is here. Um, are you satisfied with the level of the efforts or the elimination program of onchocerciasis in Africa?
I'm not satisfied with the level of elimination. We would have done better than this for this long length of time that the community that use of ivermectin has been with us. They have been with us for too long, and the disease is still there. But I think that we have some hopes. We need just more time, more education to solve the problem, as I think. And we are also now involved. It is not just sitting and waiting for people to develop the diagnostics, the vaccines for us. We are now involved in it, and I think that we're going to make a big step ahead.
That is very promising, because I ask this question because there are people who have argued that with the present dispensation of how the program runs based on the challenges and limitations, in the next 50 years, it might not be able to completely kick out onchocerciasis. Do you agree with this with this opinion? And what is as the person on the ground, the person involved in?
No, I don't agree. I think that in the next 50 years, it will be eradicated, because a lot has already been done
Yeah,
since the focus on onchocerciasis started. Till today, we've not even spent 50 years. But when we look, we see that a lot of effort has been put, a lot of achievement has been made. But I think that as soon as we start educating the masses, I think that they will take this treatment serious, because it just means that everybody is compliant.
yeah.
Within these 50 years, everybody is taking ivermectin and regularly as it should, and you will find that the adult one will die natural death, and then onchocerciasis will be eliminated. So I think in the next 50 years, a lot of things must have changed. A lot of
Okay.
work must have been done, and I think a lot of progress must have been made. That I strongly feel that it is possible to eliminate onchocerciasis. It is very difficult, but I don't think it is impossible.
Okay, thank you much, Professor Ghogomu. What are the main challenges and limitations that you face carrying out research in your laboratory?
Okay, the first is that is funding. Funding, to get funding is very challenging.
Yeah.
Uh, we get it, but I think we need to get more than that in order to really be comfortable in our lab. So
Yeah.
the first is funding. We don't have sufficient funding. I mean external funding. Even
Funding.
I cannot count on internal, because it is not even there.
Yeah, it's
There's no
quite limited.
yes.
That's right.
Yeah, so when I talk of funding, I mean external funding. It's very rare, and considering that these are diseases, onchocerciasis is a disease of the poor, the funding for it is very rare. If you go to hypertension and diabetes, yes, it is possible to have funding on this, because it's worldwide, and but in that case, it is very, very challenging too, because the competition is
The
not too
competition
easy.
is quite high. Yeah.
Guys, yes. So that those are the problems. And then we also have a problem power outage. Quite often, you have you furnish your fridge with reagents, and they cut light for 3 days. You see that the fridge is down. We have to use generators in order to power our fridges, and it's so difficult. Generators are very expensive to run.
Yeah.
And another thing is water. Water at times is not regular, so but not as current. Case of water is there, but not as that of current. At times we have problems having water. So another problem which we face, well, it relates to funding, because we find it at times difficult to keep our PhD students since there is no funding. When they stay there 1-2 years, you find them, oh, they have gone, they are somewhere else. And, yes,
Yeah,
so
I understand.
to really have a PhD from start to finish here, it's a hard nut to crack. I bear with them, because if these are people who are mature, they find it difficult turning back to their parents and telling them that give me this. So they try by themselves, and when it's taking a long time, they abandon and leave. So these are the main problems which we have. I could also add time. The more you grow, the more you leave from the assistant lecturer, growing, moving to lecturer, associate, and professor. Now you have other administrative duties, and it's a time problem, which you may not have much time really to follow up your students. So, well, we try it, but we've groomed many people who help us also.
Yeah.
Support.
That is great. So looking back, if you what are some career advice that you can give to so the question is, what is the career advice that you give to a bachelor student or a young person who is interested in scientific research?
I think that I will advise them, mostly those who are already young. Even PhD is young.
Yeah.
Yes, these are young career people. So I think that I will advise these people to request, if it is a first degree student, I think the main thing is hard work that you have to do in order to achieve your aim. And those of postgraduate, I encourage them to apply for research grants, because there are many small grants which they can apply for. Grants of $5,000, $10,000, $15,000. Those ones are for young career students. But I think we've had it with Robert, we've had it with Kabiru. They were students and they won so many grants. So I will advise them that they should not only go to the internet to exchange messages with friends, they should also search, read a lot, attend conferences and meetings and so on, learn from there, and from there they are capable of writing some short research grants and also competing in order to have some money for their research work. So for this group of people, I think that's what they can do to survive. But for the bachelor's degree, I think they need just a lot of hard work, because for them, grants for these bachelor students are not very much, or even if they exist, it's not very rare. Yes. For them, I think they can join 10 conferences, which they hear the university around. They can attend these conferences and even make short presentations, read articles, and also make their own presentations, seminars.
Yeah, it's like it's when you lay the foundation for your academic journey, indeed, at the bachelor's levels. And yeah, so Professor Ghogomu, thank you very much.